always be a lag. Good to go. You can close the doors. Now are we live? Good to go?
Good.
Okay, well, good afternoon and welcome to the Simons Electron Microscopy Center's Winter EM course. We are well into our tomography section. So you heard a lot last week from Wei Dai about traditional methods for tomography. And now we're going to be talking a little bit about the other instrument that we touched upon a little bit, but we'll go in a little bit more, and that's the scanning electron microscope. And a particular function of the scanning electron microscope, you can have a focused ion beam with it to look at volumes. And the whole purpose is, for tomography, we want to look at volumes, whether or not that's by tilting the sample or doing what we're doing here. And you'll hear a lot about that from our lecturer today, which is Bill Rice. He's our EM manager, and he's tasked with a lot of things. And he leads a lot of our tomography applications, and more so in the cryo field. And that's a specialty unto itself. And you'll learn a lot about that today. So, Bill Rice.
Thanks, Ed. So yeah, I'll talk about the FIB-SEM today. What that's used for and basic principles of it and all that. So basically for the outline, I'll just give some SEM Basics, because the course up until now has been all TEM, so SEM is quite a bit different. And then basics on the focus ion beam, application to room temperature, or what I call conventional specimens, and then using it as a cryo prep tool for cryo-TEM, and using it as a general prep tool, lift out for TEM. So sort of similar to, you have an electron beam, basically what the column is, you have electron beam coming down a column, but instead of having what we call flood beam imaging, you focus the beam actually to a sharp point. And so what happens is it comes down the column and goes through a whole piece and then hits the sample at a single point. And you have deflecting lenses so that the point itself gets scanned across what you're looking at, your object of interest. And what it does is it sort of scans back and forth, up and down across the object, and then you get basically a 2D image of it as the beam scans across. So it's kind of like doing a raster scan of a single point across the sample instead of illuminating the whole sample. And also, instead of going through the entire sample, these samples are usually quite thick. And so what you're actually looking at is basically signal that's being produced as the electron interacts with the sample and then releases either secondary electrons or backscatter electrons. If you have an X-ray detector, you can look at X-rays that are being released. But you're not looking at electrons that transmit through the sample, but rather you're kind of just getting a surface image of the sample using the electron beam. So that's kind of the main difference between this and TEM. So ideally, because you want a single point hitting your sample, you want kind of what's called a probe size. You want to have a small probe size as possible relative to the size of the pixel, and so the size of the probe is determined by the voltage, the current, and the divergence angle of it. So you sort of want it to be able to be as focused as well. You want the optics to focus to as small a point as possible on it. And so you also have lens distortions to worry about. There's spherical aberration, there's aperture diffraction, astigmatism, and chromatic aberration is voltage dependent. So you sort of have to align the instrument as well as possible to sort of get this down to a point. For astigmatism, you basically just sort of tune it as well as you can so that it doesn't look astigmatic. When it's astigmatic, it's kind of blurred out in one direction. So it's different than the TEM where you have tone rings. In this case, you're actually looking at the image that it forms itself and sort of tuning it yourself to sort of get the clearest possible image you can get. The higher, you sort of align the scope yourself to sort of get a nice image at the end.
What's like a standard probe size?
I guess it's
like microns, millimeters?
Smaller than microns, so like nanometers.
Okay.
Or hundreds of tens of nanometer probe size. So I mean, the resolution you get on this is not angstrom resolution. The best resolution you can get is, at least that we get on ours, is around 3 nanometer resolution.
Okay, so 30 angstroms.
Yeah.
Okay.
So you can't. It's hard. I mean, there are. I suppose there are newer instruments that will give better, but I mean, 3 nanometers is about the best we can get on ours. maybe, I mean, with the samples we look at. You can actually get, I'm sort of misspeaking, on an ideal sample, which is like really well conducting and sort of perfect, you can get sub-nanometer resolution, maybe about 8 angstroms or something like that. But that's with a really well conducting sample. The samples we tend to look at they're biological, so we coat them with platinum, but there's to make them conductive, but they're still not sort of perfect, and so you have some sort of it's just never so as good as TEM. Oh yeah.
What's analogous to a chrome in the TEM?
The TEM is it's as if you know when the TEM, when you look at the beam, it's kind of a flood beam, it's like a big circle, as if you focus that down to a single point using the brightness knob. So in TEM, there is some TEMs, none of the ones we have here, they run in what's called STEM mode, a scanning TEM mode. So in that case, you focus it down to a single point, and you sort of scan it across the sample, and then you see what transmits through. But we don't have STEM on our scopes that we're looking at, that we use here. It is commonly used though. So, but that's sort of what it's like. It's like you're focusing it to a single point. In TEM, there's another mode called spot scan, which is a little bit similar also, where you don't focus it to a tiny point, but you focus it to a small circle and scan that across. People used to use that on film in the old days, but I don't think it's used very much anymore. So there's the Beam-Specimen Interaction is something to consider. So what usually people do is there's various pieces of software that will do sort of a Monte Carlo simulation of how the beam interacts with the sample. And so basically the beam hits the sample, and then most of it goes into the sample, and it sort of loses loses energy as it goes through the sample. Some of it gets scattered back out of the sample. That's called backscattering. The electrons can be scattered elastically or inelastically. As you know from TEM, we care only about the elastically scattered electrons. In this case, the electrons can be scattered inelastically and lose energy, and then contribute. The sample can then release secondary electrons from it and to producing another signal. The probability of elastic scattering basically varies with the atomic number squared, and then inelastic scattering can produce secondary electrons or X-rays. And so at a very high voltage, this would be a 20 kilovolt beam, which in SEM you're usually working at much lower voltages than at TEM. It has a fairly large volume of interaction. This is going down 5 microns here. So the main volume, most of it is coming down to almost about a 5 micron depth of penetration into the sample, and it's also sort of producing a lot of signal laterally because it's hitting here, but then you're getting secondaries and backscatter electrons produced a little bit away from where the incident beam hit. At different voltages, you get different depths of penetration. So this would be the same sample, but at 30 keV it goes quite deep, at 20 keV less deep, and 10 keV even less deep. So when you want to get more of a surface view of your sample and not see too much what's underneath it, you want to lower the voltage. And so we are commonly using 2 keV or less on our scope. The Helios we have, it'll go up to 30 kilovolts, but we're generally, when we're actually doing the surface imaging of everything, we're looking more at 2 keV or less. Sometimes we do want to see what's underneath the surface in order to sort of find the sample, and then we'll be imaging up to 30 keV. There's also a strong material dependence of it. So this is same sort of Monte Carlo simulation in a heavy metal material, iron versus carbon. So these are the 0.5 micron scale bars. So it penetrates quite a bit more into carbon than into iron. So that's also a consideration. If your sample is made more of light elements, you can image it at lower voltages. You need to image at lower voltages if you only want to see what's at the surface versus what's underneath the surface. And then this equation sort of gives you sort of a rough an idea of how far it penetrates. So it basically varies with the atomic number, and then the energy of it. basically the voltage of it to the power of 1.67, and then almost linearly with the atomic number. So if you want to damage what's underneath the surface, if you lower the voltage, this can have a strong effect on how far it's penetrating into the sample. So that's something to consider which we'll get into later a little bit when looking at, say, beam sensitive samples like a cryopreserved sample. If you care about what's beneath the surface, you would want to be imaging at as low a voltage as you can get away with. So I'll talk a little bit next about the two types of electrons that we can detect on the detectors here, which are backscattered electrons and secondary electrons. So backscatter electrons basically means it hits the sample and then bounces back towards the detector, back outside of the sample. Whereas secondary ones are made more from inelastic scattering, where it hits the sample, deposits a little bit of energy, and where it hits, it releases a secondary electron. So backscatter electrons, they give a contrast between light and heavy elements. So this is a graph of the fraction of electrons that backscatter at various voltages, and this is atomic number on the x-axis and how much is backscattering on the y-axis. So as you increase the atomic number, the backscattering increases, not quite linearly, but according to this sort of curve. And it's not very voltage dependent. So this is data from 5 keV all the way up to almost 50 keV, and it's virtually the same amount. So you don't need, so the backscatter dependence gives you a nice elemental contrast, and it's not voltage dependent. On the right is a backscattered image of a stained biological sample. Sorry. And so this is a tissue sample that's been stained with heavy metals, osmium, lead, and uranyl acid and uranium. And so basically, it's a tissue sample that was embedded in a plastic resin and stained, such that all of the organic things are stained with heavy metal salts. And so what you see is really nice contrast because when you're viewing in backscatter mode, you see exactly where the metal is. It's kind of like negative stain for TEM, only the opposite, because it's sort of like a positive stain, where you're seeing where the stain is, and you can then see very fine ultrastructural detail in the cell. So if you look at when you want to look at this sort of thing, when you want to look at stain samples and see where a stain is, you want to generally be looking at backscattered electrons. Secondary electrons are less sensitive to atomic number than backscatter electrons. And so this is the backscatter curve here according to atomic number, and this is the secondary electron signal, which is pretty much sample independent. Backscatter electrons also sort of give a higher quality peak. They have a sharper peak, so they're sort of more emitting where the probe hit versus outside of the probe area. So they sort of give they'll give you a finer detail of what's in the sample and where it is versus secondaries, which are sort of more spread out. What secondaries do give you is very nice sort of topographical information. They give these very spectacular 3D views. So this is an ant sample that we took a picture of. So what happens is the signal you get depends very much upon the angle at which the electrons are hitting the sample. And so because this sort of ant head has a lot of topographical detail, the electrons are hitting it at all different angles because of the shape of it. And so you get what looks like very much like a 3D picture of it. So that's why you get these really cool pictures from an SEM that you don't get in a TEM, because you're getting this sort of 3D type information when you take a picture of it. So most of the pictures that you see of SEMs, when people look at bugs and things like that, they're looking at secondary electrons, and you're sort of seeing you're not getting very high resolution as we think of high resolution, but it is very much magnified, and you get these very fine details. You can see the sort of the hairs coming out of the ant head and the two antennae, and you get these very nice pictures of it. What you do have to do in order to look at a sample like this is it has to be conductive. So what you do is you sputter coat it with platinum or palladium, and then so that it's conductive first, and then you can get this. Otherwise, if you don't make it conductive, it'll charge up, and you'll have all sorts of charging artifacts, and it won't look very good.
Is the metal coating process, is that the same process as the gold plate coating process?
The gold plate coating process that we use for grids, that we usually actually evaporating gold onto it, so you're heating gold up a lot, and then it's kind of going to and landing on it. What we do is just sputter coating. So it's like it's in a high vacuum, but not as high for the gold evaporation, and then you have a charged plate, and then you in an argon atmosphere, and then the argon hits the charged plate, which has metal on it, and then that causes the metal to sputter off and land on the sample. So it's a much less fine. The grain is a lot more coarse. You can do it under higher vacuum to get a finer grain, but we generally are just doing it to make it conductive, so it's not so fine. It's a pretty fast procedure. It only takes a couple of minutes. We actually have I can show you the coder after class. We have it out there. One difference between the secondary electrons and the backscatter electrons is that they're much lower voltage than the secondaries have much lower energy than the backscatter electrons. So this is kind of an energy curve, and the secondary electrons are at the very low end of energy, and the backscatter electrons are much closer to the energy of the incident beam. So you can imagine as the beam's hitting it, and then the electrons are bouncing right back. They're either inelastically scattered, or they're only losing a little bit of energy and coming back. And so the advantage of this is you can separate the two different types by putting a charge ahead of your detector, so that you could then filter out the low voltage secondaries. So sort of by definition, the secondaries are less than 50 eV in energy, versus say a 1 or 2 kilovolt beam, and whereas the backscatters are going to be a lot higher. And in fact, most of the secondaries are less than 20 eV. So getting into detectors a little bit, so sort of the standard detector that's used in virtually all SEMs is what's called the Everhart-Thornley detector, where you kind of have, it's not like it's different from what we use in TEM, which is a CCD array, where the detector is a defined size. Instead of that, basically you have sort of in a way, sort of a 1 pixel detector. So the beam is hitting one part of the sample. It's emitting secondary electrons and backscatter electrons. They're then collected into it, and then hit a scintillator that converts it into light. It goes through a photomultiplier tube, and then is amplified. This says it's going to CRT density. Actually, it'll then go into a file or something, and then it then will hit the next. The beam will then scan to the next point, and then it will sort of integrate, collect the signal from that, and then move it sort of 1 pixel over to sort of generate your image pixel by pixel. So it's sort of dwelling at a certain period of time on each pixel. This could be in the microsecond level, or even less. The beam is hitting it. It's collecting the signal, and then it collects all of that intensity for that pixel. So it's not sort of a coherent imaging. It's sort of pixel by pixel imaging as the beam is scanning across the sample. And in front of the detector, you can put a charge here onto this collector, so you can make it a very positive charge and kind of try to pull everything in, and then that way it'll get all the secondaries that they might not be hitting it exactly, but they might be coming near it. But if you put a charge on it, it'll sort of pull them in to get all of the signal. or you can put a negative charge on it, say minus 100 eV, and then that'll get rid of most of the secondaries because they're 50 eV or less. So they won't be pulled in, but the backscatters will be strong enough to still get a signal. And so from one detector, you can tune this to see either full signal or just secondary or just backscatter electrons. Like I said, for the point of getting only backscatter is if you want this sort of if you want just elemental signal, what's in it, versus getting topographic information. Oh yeah, so yeah, you can apply like I just said, you can apply bias to the detector. Negative bias will get only backscatter. Positive bias will get sort of everything. There's also what's called a through lens detector. So the Everhart-Thornley detector, the normal sort of detector is going to be off to the side somewhere, but you can also get this sort of effect. You can have a detector up here where you're sort of getting signal that comes back up through the lens, and in fact you can run it. You then put the microscope in what's called immersion mode, where the sample kind of becomes part of the lens, and when you run it in this sort of mode, you can actually, with a through lens detector, you can actually get much higher resolution and better signal. So normally when we want the best resolution, we use this through lens detector, which will sort of give us this better signal. And it works sort of the same sort of way, where it's sort of not really, it's not a fixed array of pixels, but it's kind of one pixel that's sort of collecting the information, and it's sort of timed to how the beam is moving so that it generates a signal that way. So in summary, for the SEM, your image is formed by scanning points across a sample. For high resolution, you want to minimize both the probe size and the interaction volume so that you're just getting the minimum part that it's hitting. So you want to operate at low voltage, but you still need enough signal for detection. The lower the voltage, the less signal you're going to get. And it used to be, I think maybe 20 years ago, 5 keV or something was considered kind of low voltage. Now with the modern scopes, you can go 1 keV or less. Ours will go down to, I think, 300 volts. It's hard to get a decent picture at 300 volts, but at 1 keV you can definitely get a good picture. Less than 1 keV is a little more difficult depending on the sample. You can apply a negative bias to detect mainly backscatter electrons. Again, the backscattered imaging gives elemental contrast. Secondary gives sort of more signal in topographic images, and the through lens detector can give better resolution. You can also get dedicated backscatter detectors, which will work better, really give also the best backscatter resolution. Those can be a little more difficult because sometimes they can be quite sensitive. Sometimes they have to go sort of in and out of the sample as you're imaging it. So you're sort of, when you want to go into backscatter mode, you have to shoot the detector in, and then you have to be careful of the geometry of everything so that it doesn't damage the sample and all that. The SEM is quite a bit different than the TEM in that the chamber is often pretty big. So if no one is using it, I can show you after class, but you actually open up the whole chamber and put your sample inside. So sort of how we do it. You can get autoloaders too, which we use for cryo, but there's sort of a lot more room to put a lot of stuff in there. So for optimal imaging, you have to worry about, you have a lot of things that vary. So there's the voltage, so you want to use lower voltage for less penetrance. You need high quality optics for very low voltage. You can also control the current. You also control the current. With higher current, you get more signal, but then that comes with a larger probe size. So you sort of have to balance them, how much current you want versus how much signal you get. You can also control the working distance. So basically you can kind of control how close your sample is to the lens. And normally you're working, with our standard working distance is 4 millimeters, but you can go quite a bit closer, down to 1 millimeter if you want. Depends on the sample, so you can decrease the distance from the lens to increase the signal you get. There's field of view. You can control the field of view or magnification, and that gives you a different pixel size. So even though the way it scans is you have maybe 3,000 pixels across, and then you have to consider what the field of view is to work out what your pixel size is. So a typical pixel size might be a few nanometers. There's also, you have to consider the dwell time per pixel, so that can also give you more signal. So if you sit on it for 1 microsecond versus 30 microseconds, you're going to get, 30 microsecond, you're going to get a lot more signal, but it's going to take you a lot longer to image. So if you really want the best image, it might take you several minutes for it to actually scan across your entire sample. But if you don't care so much, or you want more of a live view, you want it to sort of be updating every few seconds. So these are all things that have to be considered when you're sort of setting up your imaging parameters. So you have to, if you really want the best picture, it's going to take more time. And also you can also have it do different number of scans. So it can do like a full scan of it and then another scan of it, and sort of add those two to get again to increase the signal. And then there's different detectors. I talked about the Everhart-Thornley detector and the through lens detector. We also have what's called an ICE detector, which is detects ions and electrons depending on the mode of it. And they can all be operated in secondary, backscatter, or mixed mode. And you can also get, if you really want the best, you can get a dedicated backscatter detector, which goes very close to the sample. For biological imaging, again, as you know from the cryo work, biological specimens are mostly light, so there's almost no elemental contrast unless you stain them. And the standard procedure is to stain with heavy metal salts, lead, uranium, tungsten, osmium, and look at the stain. And this is a pretty long procedure, so you have to, I'm not going to get into that at all, and because we mostly concentrate on cryo work, but what has to be done is you have to fix whatever tissue you're looking at, so you sort of fix it in formaldehyde or something similar to sort of get it stable. You have to remove the water from the sample with organic solvent, and then you have to infiltrate the sample with resin. The reason for getting rid of the water is because so you can infiltrate it with kind of a plastic resin so that you have something to polymerize the sample in. And then you have to stain the biological components with these sort of nasty metal stains, polymerize the resin, and this has been worked out, I would say, over the past 50 years. The original EM, a lot of the early pictures were done using this technique. It sort of went out of fashion for a while, but with the FIB-SEM has kind of come back into fashion in the past 10 or 15 years or so. And there are many protocols worked out for different cells, tissues, and organelles. And so that, depending on what you want to enhance, there are different protocols. So some protocols, if you're really looking for mitochondria, they'll sort of expand the mitochondria before they're stained, and so that you can kind of see the cristae and all the fine details. But if you're looking for other elements of it, you'll maybe enhance other things. And so one disadvantage is you're kind of not looking at what's directly there like you do in cryo, but you're looking at what's been stained and fixed. So there can be a bit of a worry that you did it right, that you didn't cause some sort of deformation of the cell or something like that. So you do have to be careful of that. But you can get extremely fine ultrastructural detail from this technique. So from the SEM part, I'll jump into the focused ion beam. Unless there are questions right now about SEM before I go into it. So the focused ion beam it's kind of like an electron. Except for imaging with electrons, you're imaging with ions. And what you actually have to have is the way it works is you have kind of a reservoir of liquid metal. And then it sort of flows over a substrate and forms under a high magnetic field what's called a Taylor cone. So it comes to a very sharp point, and then it kind of gets extracted out by high voltage, and then hit the sample. Gallium right now is the most popular ion source. There are other metals that are used, but gallium is what we have, and it's kind of more commonly used. So it's liquid at room temperature. The sources last fairly long, say from 500 to 1500 hours or longer. It's compatible with high vacuum, and it's a large ion for sputtering. So this is what it looks like. So this is the reservoir of liquid gallium in here. and this is sort of the metal point where it forms a Taylor cone, so it sort of flows over here and forms a very sharp tip at the end of this tip and gets extracted out. And it does unfortunately, even though it's long lived, it does only last, say, 1,500 hours or 2,000 hours of operation. So one concern of using it is a little bit expensive, because I would say we have to replace our gallium sources about every 3 to 4 months, I would say, with our standard operation. And because it has to you sort of leave it on while you're using it, but it has to form because it has to form this Taylor cone that eventually degrades. The sample has to be sort of heated every now and then to sort of keep this stable or reform the cone. Or if you turn it off, it has to be reheated. And every time it gets reheated, it uses a whole bunch of metal from the reservoir to coat it, and that's about 2 days of operation. So usually when you're using it, you sort of leave it on the whole time during the week, and then if you're not going to use it on the weekend, turn it off. You just put it to sleep so that it's not being used, and then hopefully the next time you use it, it'll start up. But that is a concern. Is that a little bit of a concern? Is that can be a bit expensive. So you have for the ion column, you have a liquid metal ion source at the top. You have focusing lenses. You have electrostatic lenses to sort of focus them really quickly and well. There's a patterning board, which is sort of similar to what happens for electrons, electron beam and an SEM. So you can similarly to the SEM, you focus the beam to a point, and then you can scan it across your sample. And it has very high speed blanking, because you want to be able to prevent milling while it's blanking. So what you can do with the ions is they interact much more strongly than electrons with the sample, because they're actually, they're gallium ions, not just electrons. So they actually have more mass, and they'll sort of hit the sample a lot harder. So with the ion beam, when it hits the sample, it doesn't penetrate nearly as much. The penetration depth is only, say, on the order of a few hundred angstroms versus microns for an electron beam. And when it hits the sample, a lot of stuff can happen. So it can hit the sample and get sort of deposited in the sample, or it can actually sputter off parts of the sample. And that's what we actually use it for, mostly is for cutting the sample. So it'll hit it and then knock off a little bit of the sample, release clusters. It'll also release secondary electrons, so it can produce a signal so you can see where it's hitting. You can actually use it to image your sample, although it's extremely destructive imaging. It'll release photons, individual atoms, and then positive and negative ions. But the main thing is it hits sort of, because it's a massive thing, it hits it a lot harder, interacts only very close to the surface, and is much more damaging than electrons. So yeah, you can get basically the sample, the incident ion beam hits your sample. It can actually deposit into the sample, and that's called doping. So sometimes people in the electronics industry might use that to sort of dope their sample a little bit with gallium or whatever. There's a lot of, you can get polymerization of, depending on what your sample is made of, it can cause chemical reactions and cause polymerization at the surface. And besides all the sputtering, there's basically a lot of chemistry that's going on here. In our group, we're not really concerned so much about this. We're more using it to sort of blast away or cut pieces of the sample out to sort of use it as a shaping tool. But there's a lot that's going on here, and depending on the lab, they'll be using the ion beam for different things. Depending on where you're hitting it, you also get different behaviors. So when you hit the sample, it sort of it causes the sample to sputter, and that can go out in different directions. If you're doing it at the edge of the sample, it's a much more efficient sputtering, because more of it's going to be released completely from the sample. Whereas if you're doing it kind of in the middle of the sample, you get a lot more of what's called redeposition. So it'll sputter it out, it'll go flying out here and then land back on the sample again. And so that can actually limit how deeply you can cut into the sample, because eventually as the sample, as it kind of cuts a trench here, more and more of it as it hits the bottom will, instead of coming out of the sample, will sort of deposit on the walls of the trench that it's cutting, so you can only sort of cut so deep. Under the standard setup, the electron beam is coming straight down the column, and then the ion beam is usually off to the side in the FIB-SEM at an angle. So on the FEI scopes, it's coming at an angle of 52 degrees relative to the electron beam. So if you want to cut directly into the sample, you sort of tilt the sample up so that it's orthogonal to the ion beam. So you sort of mill in here, and then the electron beam will be sort of hitting your sample at a non-perpendicular angle. You can also, with the ion beam, you can deposit say metals onto it. What's commonly used is platinum. It's basically an organic. It's platinum bound to this organic compound. It sort of warm it to you warm it to so that it's released as a gas and sprayed over the sample with the needle. So this is what's called the GIS, the gas injection device. So you sort of release platinum gas here. You hit it with the ion beam. The ion beam hits this and breaks apart this organic compound and releases free platinum metal, which sort of deposits right on your sample. So you can sort of protect the sample with platinum, or provide a hard surface for milling using this platinum gas. And there are other gas chemistries you can use as well. So for resin embedded tissue, basically, if you want to, you can use this to sort of get a 3D reconstruction of a large section of tissue where you prepare tissue, embed it in resin, stain it with osmium, uranium, or lead. Coat that with a thin conductive layer, and then put it into the microscope, and image with a low voltage, so you're only imaging the surface, and you image under conditions where you're just getting backscatter imaging. So this is sort of what it looks like in the microscope. You have a sample embedded in resin, and it's sort of been cut away. And you sort of, what you do with the ion, this is looking down with the ion beam. And so what you do with the ion beam is you find an area of interest, and you cut a trench ahead of that. So this is what's been done. This is a trench ahead of it, and then you cut some side trenches. And what you want to image with the electron beam is this face here coming up. So you sort of have an area that you sort of protect with platinum. That's this, and that's shown here. You trench out ahead of it, and then you want to view it kind of at an oblique angle with the electron beam. So if you remember, the electron beam and ion beam are at different angles. One's coming down, the other's 52 degrees to the other. And so in the microscope, this is how it looks. This is an area it's been protected with electron it's been coated with platinum. A trench has been dug ahead of it, and then there are side trenches. And this is looking at the same area with the electron beam. So with the electron beam, you're sort of viewing it at an oblique angle, so you can see this face of it that's been produced. So this is looking at this face here at an angle, and this is how it looks inside the microscope. This is the sample. This is the electron beam, and then this is the ion beam coming at an angle. So what you do is you cut this. You sequentially cut the face, and then you image the new face, and then you work your way in, and then from that you get all these different slices, and from that you get a tomogram of your sample. And here's an image of an actual sample that a little movie of a sample that we imaged. This is viewing it just with the ion beam. So the ion beam is going to start cutting here and work its way in. Oop. And so it sort of cuts in maybe 10 or 20 nanometer slices, and each of these slices it will then image with the electron beam, and then you get a 3D reconstruction of this entire volume. So this is sort of vastly sped up. This probably took 3 to 5 days to cut that far, but
What's the depth right there? Like, what is that scale about?
I would say I don't. This is probably. I would estimate this is probably 20 microns across here. Like that. I don't have unfortunately I didn't put a scale bar
No,
on
it's fine.
this.
So you're going in about 10 microns up?
Yeah.
It doesn't matter how deep in the Z.
Because you can just keep going, right?
Well,
Oh.
it's longer than the field of view, but it ends up
Yeah.
being further in.
You're cutting in very far here.
Got it.
So that's why it looks like a very deep hole. I could play it again, but yeah, it's sort of cutting in this way and working its way. You cut these side trenches to sort of give it a place to go, because as you cut this face, some of it's going to sputter out, and so this sort of gives it somewhere to land. Instead of making, if you don't have these, a lot of it will hit these walls here, and then those walls will get narrower and narrower as more and more stuff deposits on them. And this is what you get viewing it with the electron beam. These are 20 nanometer slices, and so these are myelinated sheaths here. You can see mitochondria coming in and out, or bits of the individual sections of mitochondria. They were looking for synapses, so if you look I think there are synaptic vesicles in there that just went by. So this is a sort of volumetric information you can get. And again, I would say this is probably 20 microns across, something like that. I didn't put a scale bar on this, unfortunately, but it's around 20 microns across, I think. Or maybe 15. Field of view is our typical field of view. I would say is 10 to 20 microns. We can, if you want to view in the highest resolution mode, you can view up to about a little less than 40 micron field of view across. But what you get is if you're really looking at ultrastructural information of a tissue or a bunch of cells, you can actually get quite a bit of information from this. And you're not getting atomic resolution, but you're getting nanometer or a few nanometer resolution, which depending on the question you're asking can often be good enough. But if you're looking at mitochondrial organization or nuclear organization or depending on what you're looking at. What you're not going to be able to do is sort of do atomic type things. However, we do have some people who have used have antibodies which are labeled with gold or something like that, and then bound the antibody to their sample, and then they can they sort of see where the gold particles are, and then they can sort of detect that sort of level of resolution. So the ideal workflow for this is that the sample's already stained and embedded. You do a thin slice for an overall map to make it easier to find features, because when you're looking at your sample, when we look at the sample that's been embedded in plastic and stained, you don't see very many details. You can kind of see where some features are, but you can't see what's beneath the surface until the surface has been cut. So finding the area of interest can sometimes be difficult. The face of the block should be polished, so it should be a very flat face that you're looking at, usually with a microtome. And again, if they've already cut off a little piece of it, maybe you make a very thin EM section on a grid and do a map of that, and then you can kind of say, oh, this is the cell I want to look at, and you can kind of figure out, we would sort of figure out where that is in the sample itself, or using light microscopy and maybe some labeling, you can figure out where the sample is. But that can be a challenge, is finding, if you want a very particular area, that can be a bit of a challenge finding it. The setup for slice and view takes about a day, so you have to sort of prep the sample, put it in the scope, Find the area of interest, mill the trench, do all of the treatment, pre-treatment before that, and then you can collect slices, and generally that takes, I would say, between 1 and 5 days, depending on how far you want you want to go, and how much of a volume you want, and the quality of the image. So if you're willing to sacrifice quality over quantity, you can go a lot deeper and do a lot more. But if you really want the highest quality image, so it looks like almost like a TEM image, then it's going to take a little while. Aligning and processing the slices to kind of make the sort of 3D movie, that doesn't take very long. It's a day or less. You can use iMod or Amira. But then the segmentation can take weeks to months if you're doing it manually. Because if you can imagine if you wanted to sort of draw out, even though this image looks really good, it's still a little bit noisy, and often what you do is you sort of draw out the features of interest. So someone might want to draw out all of the mitochondria in this. And if you're doing this by hand, it's very tedious and time consuming. So that can take a long time. What's come up in the past year or two is there's been more automation of this sort of thing using neural networks, and that's actually been a feature of EMAN 2.2 that's been introduced in the past couple of years. That seems to work pretty well. I think Micah has used it for this sort of thing.
Yeah, we should have a paper out soon that features a lot of segmentation and used EMAN 2. Works really well.
Yeah, so
Much faster.
I would recommend if you're going to do this sort of thing, and you definitely look into this sort of automated segmentation. Even if it's not perfect, it'll get you a good starting point.
And then it's easy enough to drop it into something like Amira and then manually clean things up from there if you need to.
And we have a license for Amira, the Amira software here. If any of you are wanting to use it, you can use Amira here. So now I'll get into a little bit of using it for cryo prep, which is kind of more of a newer thing. It's not that new anymore. People have been making lamella for a little while, but the idea is, if you want to image a whole cell or part of a cell, it's, as you know from the TEM part of the course, is too thick. You can't see through a several micron thick cell. It's not going to work. But what's been developed since 2013 at least is the idea of actually using the FIB by you put your frozen sample into the FIB-SEM on a cryo stage, and then using the ion beam cut out what's called a lamella, a very thin layer of the cell. So you sort of use the ion beam to cut above and below, and you sort of leave just this thin lamella, this thin slice of cell, put that back into a TEM, and then do cryotomography of that. You would tilt along this axis so that these walls don't get in the way, but you can then get a 3D reconstruction of what's in the middle of the cell. And this is quite a procedure to do, but we do have it working. The main challenge actually milling the lamella, once you've done it a few times, it isn't that difficult. You sort of have a protocol to do it. The main difficulty is getting the sample getting the cells on, making sure the cells are on the grid, kind of in the middle of a grid square, and getting it in the microscope without contaminating it or heating it. Finding the area of interest is a tough thing. So if you want to, what the cells do just kind of look like blobs on the grid, and you have to view it at kind of an oblique angle. So finding the area of interest to mill has been quite a challenge. Can be quite a challenge. Yeah.
2 questions from the YouTube channel. Can you please specify, do you take images with Amira, or iMod is the version of built-in set image collecting script? Does that make sense to you?
It's mixing 2 things.
Yeah. So the image taking is not through Amira. The Amira and iMod are sort of post-processing programs. So iMod, I probably should call it eTomo, but so Cryo-EM is an imaging program that David Mastronardi writes to collect tomograms on a TEM. iMod is his program that he uses to align tomograms and do segmentation. And Amira is another program that can be used to align tomograms and do segmentation, but that's all post-processing.
Okay. And so then how was, I guess, the images taken then? Just
So the images, it depends. So the images in here, so for these images were taken in the FIB-SEM using what's called their Slice and View app. But the images are basically taken using the microscope software on the microscope, and then they're aligned using IMOD or Mira.
And that software is called Slice and View.
The imaging software on the FEI is called Slice and View. Yeah,
Okay.
I'm sure Zeiss has their own version of the same thing.
Okay.
Joel probably has the same version of the same thing.
And then a second question, in the next slide, how was the cell dehydrated?
Oh.
Next one. Next, that one.
Oh, and the cell actually, this actually should be, there should be water around here. This is just like a diagram from the paper. The cell's not really, basically you have the cell grown on a grid. You'll sort of freeze it by blotting using the standard technique of, you know, a Leica or a Vitrobot, so you blot most of the water off, and then plunge freeze that grid, and then you sort of are left with the cell with a little bit of water around it.
Okay.
But it's
So
not.
it's still hydrated.
It's still
right?
hydrated, yeah. It's not dry.
Got it.
It looks dry here, but yeah, it's not really dry.
Okay. Continue, please.
Okay, and then here's a picture from their sort of review paper that describes how it works. So this is how it looks inside the microscope, where you mill this is inside the these are pictures taken with the electron beam inside the SEM, where the ion beam is milled here, and this is sort of the edge of the cell here and here, and this is the thin lamella that was left. And there's another thin lamella that was left here. And then these are tomograms. These are images from these are TEM images taken of this lamella. Actually, these are probably slices of a tomogram taken from this lamella. So the grid was after being cut like this to leave a thin lamella. The grid is then transferred cryogenically into a TEM where you then find the same region of interest and do tilt tomography of that, and do the reconstruction of a tilt tomogram, which I think was covered in last week's lecture by Wei Dai, right? Yeah. And then you can then get very, very, very fine details. So the standard setup that we have here for the Lamellae preparation is, you basically have to have you have your grid frozen. You have to clip it, which we use using the auto grid loading station, and we have a specialized shuttle from Quorum, which will hold up to 2 clipped grids. It's got the grids go in a little slot, and then the slot is covered by a little shield to sort of protect it from the atmosphere. There's a loading station to put it into the FIB-SEM, and then it sort of goes through a bayonet system into a cryo chamber, and then from there into the microscope itself. And this is these are images we took here a few years ago. So once you cut the lamella, you sort of they're pretty easy to find. So this is taken from a grid atlas, and these are the thin lamella that were generated. So the ideal workflow is you grow your cells on a gold grid. You can't use copper grids because the copper is toxic to cells, and then plunge freeze them. Ideally, you could image by cryo-light microscopy to find features. Load into the FIB-SEM and mill slices. That's about a day's work. In a day, I think Alice can do 8 to 10 lamella. Full day.
It's happening.
Yeah. From there, once you take it out, you can then keep it stored at a liquid nitrogen until you get TEM time. You load it into the TEM, collect tomograms, 1 to 2 days. We can do up to 50 tomograms a day, I believe we're doing right now.
Collection?
Yeah.
100.
100.
24 hours.
Align and process them using the Protomo or IMOD. Takes a day, and then again you do have to do the segmentation or subtomogram averaging. This will be the long step. Although, to be honest, all of these steps are sort of hard. Freezing the cells well can be hard. You have to get them to grow well on the gold grids. We have a cryo light microscope at NYSBC. It's kind of not ideal. It can work for basic things. We're looking into a better solution. Loading into the FIB-SEM, that works pretty well, and the milling can assuming all of the equipment is working well, that works pretty well. Rescuing it we can do, but it is a lot of work. What we're also looking now actually is doing sort of the slice and view type imaging, using the FIB but in cryo mode. So you actually can do sort of slice and view of an unstained sample in frozen. It's been shown by a couple of groups. This is an example of one of the groups where they basically took some cells. I'm not sure if it was cells. Actually, it was C. elegans. It was one of the samples they looked at, but they high pressure froze it and then put it directly into the FIB-SEM, again under cryo conditions, keeping it cold, and then ran the slice and view type software, imaging at a low voltage. They used 2.33 kilovolts, and they actually were able to see pretty good contrast. So this is an unstained frozen sample, and this is a sort of an example of the type of contrast that you get. And this is compared with the type of contrast you get from stained. So stained, you get a lot more contrast. It looks a lot better. But the advantage of this is you can actually you can do this unstained. So you go right from freezing. You don't have to worry about all of the staining and embedding into plastic resin, and you can actually directly get kind of volumetric information. Potentially, you could also use this if you wanted to sort of find a region of interest to sort of cut out. And so we're investigating this right now. Another thing you can use the FIB-SEM for is what's called lift out. And in that case, you basically want to, that's been done in the material science world for many years, where you sort of have a sample, a bulk sample of something. You know, maybe it's something from a piece of metal that failed and you want to study metal fatigue or the fine crystalline arrangement of it in TEM, but it's too big. So what you do is use the FIB-SEM to sort of cut out a small area of interest and then a small volume of interest and then put it onto an EM grid. And so what you do is, the sort of standard protocol is you have this bulk sample, you have an area of interest that you want to look at, or it could be anywhere in here. You protect it with a little bit of platinum, and then you mill a trench around it. This is again the piece you want to see. So using the focus ion beam, you cut here and here, and then this is the top view of the trench. You then sort of polish it, so you cut it very finely at a lower current using the ion beam, so that you can start seeing details with the electron beam of what's actually in it. Do some more polishing to make it very thin. So this is now the area of interest has been thinned to quite a small region. This might only be less than a micron. This is sort of here's an oblique view of the same thing. You then cut around it using the ion beam to sort of cut almost all of it off, but leave a little piece attached to define sort of the area for taking it out. Again, here's the top view of it. And then you thin it to make it very, very, very thin, to be electron transparent in TEM. And so now the sample is extremely thin. It would be thin enough to look at if you were looking at it on the side view by TEM. And so using a micro manipulator, you attach it to what's called like a micro manipulator. Ours is called an omni probe. It sort of attaches to one end of it. You sort of glue it to the omni probe using platinum, and then you cut away the last piece, and then you move this, and then move that onto a grid, and then from that you can do very thin section imaging of it. And so there were actually, there's been a couple of groups who are applying this to cryo. The first was sort of the Jürgen Plitzko's group with Julia Muhammad has been the main person doing that. And so she came out with a FIB milling and lift out approach for cryo work. So they did that again on C. elegans tissue, where they managed to freeze C. elegans onto EM onto I think, I'm not sure if they froze it onto EM grids or how I can't remember exactly how they froze it, or it might have been it was high pressure frozen, so it was high pressure frozen onto a grid, and then they did the same sort of lift out procedure where they sort of cut trenches of it, and then using a Cool-Thornley probe, they were able to lift it out and then put it onto an EM grid.
So 2 more questions.
Yeah.
From Lita. I think Alex has answered one. So does your setup have a port for installing an EDX lens? EDX detector, sorry?
We do, but we don't have an EDX detector.
Okay.
Yeah.
And then have you done lift out on frozen at our place?
We're trying to do that. We're
Okay.
getting that working, actually.
Right.
So we have almost all of the materials in place for it, but we're just a matter of doing it. And that's taking quite a while.
Thank you.
You need in order to get this to work. Oh yeah, here it looks inside this. This is one of their TEM images. So they did see a lot of curtaining. They looked at the diffraction of it. They didn't see any evidence of ice diffraction. So, but they were able to see a lot of fine details of it. So this is sort of at the edge of what you can do. The equipment needed for that was quite a bit. They needed basically they needed a cooled micromanipulator, a gas injected device to sort of glue it onto it when it's cold. You need a special copper cooling braid to cool a micromanipulator. You need this is like a lot of equipment to get it to work. And then you actually put it onto these sort of half grids where you can sort of stick it on there. This is another example of another group from actually in Albany where how they did cryo lift out, and then they did the same sort of thing where they cooled it and then glued it with water in this case, and were able to lift it out. So this is sort of the idea. Again, you sort of find an area of interest, dig trenches in front of it. They used ice to deposit in front of it. They used ice to sort of glue it onto or water ice to glue it onto the micro manipulator, and then move it onto a grid. And that's all I have to say about this today. So.
One request: Can you re-show the video where it's the slice and view when you are going up those 10 nanometer slices?
Oh yeah, sure.
If you could just play it like a couple times.
Yeah, I can leave it on it for a bit.
Cool. Thank you.
Oh, I hit escape. Maybe I shouldn't have hit escape. Okay.
It just takes a second.
Oh,
Yeah.
okay. There. This one, right?
I believe they missed the entire slice and view demo.
Oh.
Yeah. It's either that one or the one before that. I suspect she might have been talking about the one before that.
Okay, so this is what you get from the slice and view.
Yeah, the results.
It's going back and forth from using the E-beam imaging, and you get the sort of this is like basically a movie going through the tissue. So these are 20 nanometer slices. And if they want to see what the ion beam did, I can go to the previous one.
Yeah, just like 2 times. These
Yeah.
are recorded too, so she can go back and
Okay.
look at that too. That's great, thank you.
Yeah. And this is the ion, the view of the same thing from the ion beam. Oops. Where it's cutting through. Again at 20
Yeah.
20 nanometers at a time.
That's great. Okay, thanks.
Bill, for the cryo-FIB
Yeah.
samples, how do you deal with like heat damage? Or is that something you just have to slow the pace of milling down?
Yeah, you want to mill when you're doing the bulk mill ahead of it or something like that. You can mill at a higher current, but when you get closer and closer and it's getting thinner and thinner, you have to lower the current, and it takes longer and longer. But fortunately, ice, it's so light it doesn't take too much to mill through. So I think, what's your smallest current? Are you using 20 picoamps, Alex, when you're by the end?
I don't remember.
I think around 20 picoamps is the smallest, and you might start.
Yeah, around 20. You're right.
Yeah.
I just wasn't confident with the units because you kind of ignore them at some point.
They get
Yeah.
you down to like 2 kV for the ion column?
The ion column we actually always run at 30 kV.
You do?
Yeah, I think there's no reason for us to lower the current of the ion, the voltage of the ion column. But yeah, we haven't detected any, and there've been several. I think anyone doing it also has not detected any crystallization of it, so it seems not to heat it too much when you're cutting it.
Like material science, if you're higher ones, they get the gallium redeposition all over the place. I don't know.
Yeah.
Biological material is different, and it necessarily won't adhere to it.
Yeah, we haven't really. The first time I did it, I saw some redeposition of like little blobs of stuff on the surface of it, but I thought I might have been doing it too much, and I think I was hitting some of the grid bar or something like that, and it was sputtering copper onto it. I don't think we've seen any. I mean, there will be some sort of doping of it, or penetrance of gallium into it, but there will be sort of individual things. I don't think we've ever really seen blobs of gallium or anything like that. But again, when we're getting to the fine milling, you're usually doing it at a pretty low current.
Why is there a curtain effect on thin slice?
Oh yeah, that's a good question. So sometimes you can, if the surface isn't very even, or it's hitting it, if the ion beam when it's cutting, it hits things at different densities, it won't cut as well, and it kind of leaves a mark going down, which we call curtains. With the ice, probably it wasn't. You can't really. I think the ice probably wasn't very smooth or something when it was milling it, and it's kind of hard to sort of deposit platinum onto it very well. So I think that can cause a curtaining effect of it as it cuts. It sort of leaves a sort of surface mark on it. We see that sometimes with the slice and view. That's sort of why we use platinum to protect it. So before you do the slice and view, you coat this whole thing with a layer of maybe 1 micron of platinum, and then it's sort of like if you're slicing bread, it's easier to slice it if you have a hard crust ahead of it versus if it's just soft, it doesn't cut quite as well. It's kind of an analogy. Okay. Okay, well,
Thank
thanks.
Everybody sign the sheet?
Nope.
Floating around somewhere.
Oh
It's
yeah,
on the table.
Elena has another sheet, right, for NCCAT for
Well,
everyone to sign.
they just sent an email out to follow up on that.
Yeah.
How many are showing up on Wednesday for the journal club? Okay.
Cool. I think Gail is going to present on John Briggs's map of the HIV-1 capsid at 3.9 angstroms solved using subtomogram averaging. So I think we're probably going to have a FIB lift out paper, which we probably should have done first. So this will get a little bit ahead of the lecture, and then we'll be talking about subtomogram averaging before, I think, Alex gives the lecture on subtomogram averaging. So
That's coming up Monday.
should be, you'll be seasoned experts by the time we get to the lecture.
It's more of a discussion then. Maybe. All right, that's it.
We'll see you guys on Wednesday.
Thank you.
Unless you want to see a dual beam FIB SEM.
Oh yeah, if anyone wants to see it, I can bring you over there. Actually, because there's not that many people.